So, hello everyone. I am Veronica, and I'm from Mexico. I'm from the UNAM. So, the name of my presentation is Exploring Carotenoid Colors, Insight into Evolutionary Dynamics in Passerine Birds. So first, I want to start with plumage coloration. Well, plumage coloration is one of the most conspicuous secondary sexual ornaments in birds, and it may due to the microstructure of the feathers and or the pigments deposited in them. And some of the pigments responsible for most animal coloration are carotenoids. And of course, they are involved in plumage coloration too. So carotenoids usually result in bright red, orange, and yellow colorations. But they are not only used as pigments, but also play roles in the self-maintenance of individuals, as they are involved in protection, vitamin A synthesis, and as antioxidants. Also, a special characteristic of carotenoids is that birds cannot synthesize them de novo. They must eat them. And for all that and for the physiological pathways where they are involved, many of the classic examples of honest signaling in animals are based on this coloration. And because parental performance cannot be observed before mate choice, we thought that the condition of mate must be evaluated using indirect traits that honestly indicate quality. And for all the relationship with the diet, like interphagoginability and the condition, we hypothesized that carotenoid coloration in plumage could function as a signal of direct benefits in terms of investment into parental care by mates. But this is not new. Burley already proposed the good parent hypothesis in 1985 with zebra finches, but we want to see what happened on a big scale. So we focus on passerine birds. Why? Well, because passerine is the most diverse and widely distributed order of birds, and it's estimated that at least 41% of the species show carotenoid colors in their plumage. Also, passerine species extract and accumulate carotenoids more efficiently compared to other groups of birds. So we assessed the relative contribution of males, just males, in parental care, that was like nest building, incubation, chick feeding, and the presence, the binary 0 or 1, and extent, using patches all around the body of the birds, of carotenoid coloration in the plumage of 349 species, while controlling for phylogenetic relationships. We also analyzed their diet, body size, geographical distribution, and polygyny, because this could potentially confound the relationship between coloration and male care. And I want to make a special emphasis on diet. On diet, we developed a scoring system following also Owens, who score carotenoid intake based on a scale index that ranks 7 diet categories based on the carotenoid content. 1 for seeds and nuts, 2 for nectar and pollen, 3 for vertebrates, 4 for invertebrates, 5 for flowers and fungi, 6 for fruits, and 7 for algae. And we collect the diet data from the descriptions in the Handbook of the Birds of the World Alive. And we estimate how important was each group in the diet of each species of bird, and we multiply the relative importance of each diet category by the carotenoid content. And what we found? We found that carotenoid coloration is associated with decreased male investment in offspring care. That doesn't mean that carotenoids are not honest signals, just that they may be signaling indirect benefits or maybe other types of direct benefits rather than a greater contribution to the offspring care. And we found a negative association with size. That is not new. I think Cooney in 2022 found the same relation with body size and coloration in birds. But the thing I want that you notice is diet. So we can see a positive relationship here, with diet. So species that consume carotenoid-rich foods have carotenoids and also have more carotenoid coloration than the species with carotenoid-poor diets. And this can sound kind of obvious, but if nature shows itself as it is, science will not be necessary. So then we thought, so what is happening with carotenoids and fruits? So although algae are the food with the highest amount of carotenoids, there are not really many passerine species that feed on them, but there are many frugivorous species. in fact, they are one of the largest group of frugivorous. So they are diurnal dispersers per excellence. They have excellent chromatic vision, which is why it has been suggested that they use colors, especially bright reds and purples, to locate fruits to consume. So frugivory represents a mutualistic interaction, and more with angiosperms because they produce the fleshy fruits. And as a mutualistic interaction, both birds and plants benefit, with birds dispersing the seeds and plants providing nutrients through the fruits. And given the important role of plants as food resources for passerine birds, these groups have a long evolutionary history that may lead to establishing coevolutionary dynamics that influence phylogenetic patterns. So I'm going to talk just about the part of the birds because it's what I like. So I'm going to evaluate the frugivory in the diversification of passerine birds as a proxy of the mutualistic interaction between plants and birds, along with the carotenoid coloration that was my original question. And I want to see what this shows, okay? Okay, maybe like this mutualistic interaction have played a key role in the success of the diversification of the passerines. So this time we want to do it big. So we classify the diet of 5,470 species of passerine as frugivorous or not. And we ask ourselves two questions. First, is there a correlation between the evolution of frugivory and carotenoid coloration? And does this mutualistic interaction between passerines and angiosperms contribute to a higher diversification rates in passerine birds? And to answer this question, we employ a Bayesian approach to test for correlated evolution between frugivory and carotenoids. And here you have the independent model. You only have like four transitions because it's independent of each character. And the dependent that it's not as simple as change to the other one because it depends if you are frugivory or not. And to answer the second question, we employ graphical modeling in Bayesian statistics to infer parameters of state dependent and speciation and extinction. So these are bird-ended processes. And what we found? Well, our results confirmed the existence of an evolutionary correlation between a frugivorous diet and the presence of carotenoid coloration. And here, the thing that you can see is once the carotenoid coloration is lost, from C1 to C0, it's not easy to evolve it again. You can see it over here. And once frugivory, that is F, is lost, like from here to here, the ancestral state, the loss of carotenoid coloration becomes more likely. And here you can see the ancestral state graph. Okay, so you have these two with more probability that can be F0C1 and F1C1. So in both, you have the C1, like the presence of carotenoid coloration, but the frugivory remains uncertain. And our expectation was for frugivory to be ancestral to carotenoid coloration due to the requirement of the extraction from the diet. But our results don't show that. And for the second one, to answer the second question, we model transitions between the states of the traits and different rates of speciation and extinction associated with frugivory and coloration. And we also fit models that incorporate hidden states, which reduce the chance that the effect of the focal traits on diversification found to be significant when it may simply represent heterogeneity in the diversification process. And we compare this model with the character-independent counterpart, that is this one over here, to investigate whether something other than frugivory and carotenoids better explains the diversification process. And then we compared the models, and we found the best one, and that is the Mujice. And the thing that you can see here is, despite the potential link between frugivory and coloration, driven speciation, we did not find a greater diversification associated with our focal traits. We found also that the inclusion of the hidden states improves the model's fit. So these are my hidden states, these are my focal traits, and these observations suggest that there exist factors beyond our focal traits that drive passerine diversification. So at the end, I just want to summarize it in 6 take-home messages. First one, ancestral state uncertainty. The assumption that frugivorous is ancestral to carotenoid colors is challenged. The ancestral state is unclear. We also found evolutionary link between carotenoid coloration and frugivorous diet. Carotenoid coloration may have driven a dietary shift to frugivory. Birds may have obtained carotenoids from other dietary sources before specializing in frugivory, because as I said before, like you can obtain the carotenoids from all the types of diets that birds have. The thing that changes the amount of carotenoids. Also, we did not find a higher diversification rate associated with carotenoid coloration and frugivory, and we found that models can indicate that other factors beyond those tests influence passerine diversification. And well, I want to thank to all the people that collaborate in this paper, especially to Alejandro, that is my supervisor, and Rosanna, that was very kind and helpful with the analysis, and Tamas and Andras, that were my co-authors of the parental care paper, and of course to the institutions that give me the money to be here. And thanks to you for the time. Well, yeah, but I don't know in passerines. I think in passerines, the reds are from carotenoid coloration, but of course in Psittaciformes and other orders of birds, you have another type of pigments. But for passeriforms, the reds, orange, and yellows depends more of carotenoid. I haven't read anything about other type of pigments, at least in this order. Yeah. Yeah.
So for the 5,000 birds that you looked at, how did you assess carotenoids? Were you just looking at images, or how did you?
Oh, well, I based on data, from the data of another publication that assesses the carotenoid presence and or absence of all the birds. So I did use data. I didn't do that work. Yeah. And thank you. Super.
Hello, everyone. It's nice to see you all here. My name is Juliana Soto. I'm a second-year PhD student at the University of Illinois in Urbana-Champaign. And I'm very happy today to talk to you about this exciting project I have been working on. It's about evolution of bacterial endosymbionts that lives inside feather lice that lives on the feathers of birds. So it's a really cool system. So let's get started. First of all, I want to tell you that insects can establish diverse symbiotic relationship with intracellular heritable bacteria, as you can, are you seeing like, well, heritable bacteria that lives inside the insects in cells called bacteriocytes that are grouped in an organ called bacterium. They are very diverse in terms of morphological in different arthropod groups. These endosymbionts provide multiple benefits to their hosts, including protection and resistance against enemies, for example, parasites, pathogens, and predators. Others can provide physiological tolerance to insects, for example, high resistance to high temperatures, and also benefits for their fitness, for example, to have more offspring or higher survival rates. And one of the most studied and, let's say, common benefit is about nutrition, where they help insects to synthesize, for example, essential amino acids or vitamins that are lacking in their diets. So these endosymbiotic bacteria have been crucial for the evolution of insects, and they have promoted a specialization of the insects in different feeding niche. So as you can see in this graph, there are like diversity of different feeding specialization in insects, and this has repeatedly impacted the diversification of insect lineages in different ways. But one interesting question about these endosymbiotic bacteria is how they are acquired by insect lineages. This is an interesting question because it's been long thinking that these endosymbiotic bacteria establish long-term relationships with the insects. That's like the main pattern that has been studied. But we have found in the literature two main acquisition scenarios. One of them is that the endosymbiotic bacteria come from free-living ancestors. So here is a graphic where the red dots represent like the free-living ancestor, and they have some features that differ from the endosymbiotic lifestyle. For example, they have larger population size, their molecular evolution is slowly, and in a phylogenetic context, we can see kind of this pattern where they have a small branches in the phylogenetic tree. And in the other side, another way of acquire the bacteria is from other ancestors that has this bacteria, right? So we have this example of donor lineages and that transfer the endosymbiotic bacteria to the recipient. And so this will be reflected in a phylogenetic context and a more robust phylogeny with long clade nodes and short branches. Because these endosymbiotic bacteria are vertical transmitter from mother to offspring, so we would expect like to have this kind of robust phylogenies in different groups. And one very interesting group to see the evolution of these bacteria are those that are deficient some nutrients in their diets. So the feather feeding lice that we can see here, one of these insects that actually are very diverse. So they feed mainly on feathers, and the feathers are composed mainly of keratin. The keratin is rich in amino acids, but it has a lack of vitamin D. So these insects have to find a way to have this nutritional. And one of the first records of these endosymbiotic bacteria was in the feather louse genus Columbicola that parasitize doves. And as you can see here in this graph, the endosymbiotic bacteria are the ones that are highlighted in red. And this particular bacterial lineage corresponds to the Sodalis genus There is a bacteria that has been reported in many groups of insects, and some of them are endosymbionts of insects, but other species or forms are free living species. And interestingly, in feathered lice particularly, have been like 2 studies so far about the phylogenetic relationships between the lice and the bacteria. And here we have 2 examples of the patterns that have been found. In the left side, we have the Columbicula genus, when we have like a tanglegram showing the coevolutionary relationships. And it's been found that these bacteria in this group have acquired repeatedly from closely-related free-living ancestors. But if we see in the right figure, we see like a more co-speciation pattern where the lice and the bacteria have evolved through time, and it's more like the model that I show about the inheritance of the bacteria from a common ancestor. So we were very interested in to see what will happen in a broader evolutionary time scale and in a broader taxonomic scale. So we selected the big group of feathered lice that it's around 3,060 species described so far, and they correspond over to 150 genera. And as we can see here in this phylogeny, they show a big diversity in terms of morphology. Feathered lice, as I say, like live on birds, and we sample lice from different species around the world. And we were interested in to answer these 2 questions. Are the Sodalis alid bacteria that we found in these first 2 genus present through the diversity of all the feathered lice? And we were also interested in to see if this repeated acquisition pattern is shown throughout the diversity of feathered lice. To do that, we basically, very basically reduce, we sequence the whole genome of the louse species. So the advantage of the whole genome sequences is that we can catch the genome or the sequence of these bacterial lineages. So we basically meaning the bacterial sequences from the whole genome using different pipelines, and with that reconstructing the phylogenies under maximum likelihood, and we did an analysis of coevolutionary relationships. Well, some results so far is that we found that this Sodalis group is an extensive but has variable presence through the feather feeding lice. So from the 1,020 samples that we analyzed, the whole genome, we found in proportion in 22.4% that this corresponds to 57 genera. And here in this graph, I'm just showing like the frequency and the different louse genera. And regarding to the phylogeny, I include here like 2 displays of the same tree to show that this is a not very well resolved phylogeny. So we have a star topology or a comb-like topology, which indicates that there are not very solved relationships, and we don't have like very high bootstrap support. But for the basal groups, the endosymbionts of lice with the other groups of endosymbionts that we use as odd groups, we have strong relationships. We found like two major clades, the pink one that includes the endosymbiont from Sodalis glossidinus, the tsetse fly, close related with some feathered lice, but the main clade is composed by a few endosymbionts from insects and interestingly from a free-living bacteria, which is clustered together with the rest of the feeding lice. This Sodalis precautivus was reported from an infection from a human, like from a stick. So it's very cool and interesting because it kind of, it's been reported also in wood. So this like transition to the parasitic kind of life, it's also something cool to explore. Also, we found like a significant variation in branch lengths. Oh, sorry. And as I showed, like the evolutionary rate has an effect here, which supports that these bacteria, bacteria from the feathered lice are most likely come from a free-living ancestor. And in terms of the cophylogenetic patterns, we test for the overall congruence between both phylogenies, and we found that more congruence than expected by chance, so relatively few co-speciation events. And yeah, as overall conclusions, there is a widespread presence of this bacteria, but it's not universal. We found evidence about the repeated acquisition, and we found cophylogenetic incongruence between both the louse and the feathered lice. And I want to acknowledge all the people involved in this project and the funding institutions. Thank you so much.
Cool. Hi, everyone. I'm Christophe. I'm at Durham University in the UK. Today I'm going to be presenting a chapter of my thesis, which is multivariate trait evolution of a seasonal polyphenism at the continental scale. So, a lot of people study trait evolution and how the traits of species have diversified and evolved across the tree of life, but that's predominantly focused on traits that are really set within individuals, or traits that are only really one value for a given species. Now, there are examples of, obviously, variation within the same species leads to gradual evolution over time. What I want to talk about today is polyphenic traits, traits that have the capacity to be exceedingly different even within the same individuals. So the same individual with the same genotype has the capacity to develop into one or more very divergent phenotypes. So the larvae of the peppered moth can develop into a brown colour or a green colour depending on the substrate which it's laid on. The same individual with the same genotype can develop into multiple different phenotypes. Now, because we live on a planet with a tilted axis, we have seasons, so there's a periodic cycling, repeated changing on the environmental conditions that animals frequently experience. So what we can often see is seasonal polyphenism, where the pattern, the species change their phenotype over the course of the year. So this is the species I study. It's called the ruby spot, smoky ruby spot damselfly, or Heteronotetia, and it has exceedingly strikingly variation in melanization across the year. So individuals that emerge at the start of the year have basically entirely nearly clear wings. As the year progresses, individuals emerge with much darker wings, and then that falls back down towards the end of the year. Now, importantly, an individual's genotype, or the adult individual, does not change its phenotype over the course of the year. An individual emerges, it lays down its melanization, that individual lives for a few weeks, it dies, but over the year, as new individuals emerge with gradually different phenotypes, the whole average phenotype of the population changes. What I want to try and understand is, how does this evolve? because we have different populations which have very different patterns in seasonal polyphenism. Some populations which vary absolutely massively, and some populations that only vary ever so slightly in the year. How do we switch between populations that have really high levels of seasonal polyphenism to populations with very low levels of seasonal polyphenism? Is there a phylogenetic signal to this? So are more closely related populations, do more closely related populations have more similar overall patterns in seasonal polyphenism? And then also, do we see coevolution between the trait value in different seasons? If we increase the trait value in, say, the summer, do we also see a corresponding shift in, say, the melanisation or the level of melanisation in the off-peak season? So just to reiterate, there's sort of two scenarios we're trying to test between. One, that it doesn't matter what's going on in one season, in the other season, you could always just have whatever trait value you actually want, dependent on the environmental conditions or the selection for the specific phenotype that is best set in that season, in that specific population. But what we often see in trait evolution is that, traits have co-evolved together. You change one trait, you change a different trait. What would be really interesting to know is, yes, if you increase the trait value in one season, do you also see a corresponding increase or decrease in the other seasons? And that's what I'm trying to test within heteroneotetia. Now, there are some challenges with this, mainly in data acquisition, right? If we want to model or study what the seasonal polyphenism is in all different populations, then we have to go out and get the phenotype of all the different populations we want to study. But because it's a seasonal polyphenism, we also have to go to all of those different locations at every single time of the year. And for this species, it lives basically from the Canada border all the way down to Panama. That's a big ask of anyone. So you only have, we can go out to these locations, but if you only have the trait data from the end of the year, you have no idea what's going on in the middle of the year. This is the challenge that we've been trying to overcome. We think we have done this, so many people have been going out into the field, collecting this species of damselfly. I've been very lucky to inherit a freezer full of them from across the species range. We've sequenced them, built a phylogeny. Happy to go into the details of how we did that later, but I'm going to say we have a phylogeny. While people have been going out into the field and collecting the samples, they've also been taking photographs of them in a standardized format, set on a scale. We can take that, open up ImageJ, cut out the amount of melanisation from the actual wing. Lots of people over a decade have been doing this, going out into the field, but that still, even now, does not include all the species range at every single time of the year. So we've been trying to fill in the gaps. How we've done that is download basically every photograph of this individual from iNaturalist, built a computer vision model that can cut out the wings of a damselfly, and then manually from that we can calculate the degree of melanisation. We have also submitted our own photographs to iNaturalist, of individual damselflies that we also have the standardized measurement of them, so we can compare those and see, okay, are they valid measurements? And we get a pretty strong correlation between those things. So we can incorporate all of the standardized measurements into the same data set as an iNaturalist one. So, pulling that all together, that now gives us a data set of 5,615 individual damselflies across the entire species range from Canada all the way down to Panama, but also, crucially, across the entire year. Don't think anyone's managed to pull together a data set of this scale before, which is really exciting. There's loads of different questions we're hoping to answer this, but for the moment, we're going to focus on phylogenetic signal and coevolution. We can combine the phenotype data with with our phylogeny, basically based on the geographic location of those, and we can get an average value of the populations on each tip for each season. So we're just modeling basically a peak value and an off-peak value, and then from that we can look at, okay, how does this trait or how's the seasonal polymorphism evolved on the tree that we've constructed. So now we've got that combined all together, we want to ask, okay, is there a phylogenetic signal, and do the values, does the value we see in the peak value co-evolve with the off-peak value? Do they increase and decrease together, or does it matter, what thing? Because potentially, if they actually do co-evolve, it limits the ability of seasonal polyphenism to be an effective strategy for adapting to the specific environment in that season. Because if you're trying to evolve to be one value in one environment in one season, but that's also dragging over the trait value in a different season, then you can get an offset going. So potentially it's limiting the amount of adaptation that seasonal polyphenism is actually useful for. Very quickly, how we're sort of done that is use parametric bootstrap likelihood ratio tests. So we've taken a more simple model of the trait evolution with either a single selective regime or without coevolution, simulated what we'd expect given that more simple model, and then compared the likelihood ratio of what we get of the compared the likelihood ratio on the simulated data to a more complex model with multiple different selective regimes or with coevolution, and then we can compare that to the observed data of what we actually have and see if the likelihood ratio falls inside of the distribution likelihood ratio we get for the more simple model generated on the more simple model. So this is the simulated data set for single selective regime across the entire tree, but with no coevolution, and we see that our observed data, the likelihood ratio falls outside of what we'd expect under a single selective regime. So that suggests that within heterogeneity, we do see different selective regimes where the patterns of seasonal polyphenism is being drawn to our different local optima on different parts of the tree. In this case, that's a high latitude regime, and then very different regimes between the Pacific and the Atlantic. So we have heterogeneity in rivers that drain into the Pacific and drain into the Atlantic. They have very different phenotypes. So the populations that live in the Pacific have very low levels of polyphenism, and populations that are in the Atlantic tend to, although not all of them, have much higher levels of polyphenism. So some evidence to say there's multiple different selective regimes for how this seasonal polyphenism is evolving. When we compare run models of trait evolution with multiple different selective regimes, but without coevolution, then we see that the likelihood ratio for the observed data falls within the distribution of that. So this is evidence saying within heterogeneity, we don't have the evidence to say that the traits are co-evolving, that it doesn't matter what the trait value is in one season, the trait value in the other season is free to be whatever it wants to be. However, I do want to point out that the actual value we get for the amount of coevolution, so the standardized covariance, is actually extremely negative. And that's not the scenario that I was talking about at the start. I was talking about if you increase the value in one trait, you increase the value in the other. What we actually have not evidence for, but what it's pointing in the direction of, is the opposite. It's actually negative coevolution. It's that if you increase the value in one season, you actually decrease the value in the other, and if you lower the one in the other, thing. And that is essentially suggesting that actually in an interesting scenario where the way this seasonal polyphenism evolves is not by increasing or decreasing together, but it's actually just changing the total magnitude of the seasonal polyphenism, which is kind of reasonably intuitive if given an ancestral trait without seasonal polyphenism, you increase or decrease, and the fastest way to evolve to change it in one season would be to change the total magnitude overall, which again would potentially lead to the same scenario I was talking about, where it would limit the ability for each season to get to the perfect trait for that season at that time of the year, because you're still going to have those potential coevolution factors. However, we don't have evidence of that in heterogeneity, but I think it's an interesting way of thinking about how seasonal polyphenism could actually evolve over time. That's everything from me. There's a lot of people who collected out in the field over many years. Too many to thank here, but specifically my supervisor, Jonathan Drury. I'd be happy to answer any questions. And I've also handed in my thesis, so if anyone has any postdocs who want to chat about anything I'm working on, please come speak to me. Thank you. Yeah. Sorry. Yeah.
Can you rule out antagonistic pleiotropy as a simple explanation for the pattern you see? And then call it coevolution?
I don't know. It's about 2 a.m. for me, so I'm a little bit tired. Can I chat to you after? Is that all right? Yeah, I will come speak to you, I promise. So sorry. But please ask me more questions. I've just dodged that one, and I don't want to dodge them anymore. That's a very good way of not getting more questions, isn't it? Cool. Okay, thank you very much, everyone.
Well, I guess we can get started. Hello, everyone. My name is Justin Merondun. I'm excited to share the fourth and final chapter of my dissertation today. I'm extra excited to share it because I defended last month. So if you have any suggestions after, would like to chat, be happy. We're hoping to submit this within the next couple weeks. So happy to hear any thoughts you have to share. So we're already pretty primed on coevolutionary interactions, so I'm going to jump just right to a species introduction. So may I introduce the common cuckoo. The common cuckoo is particularly infamous because it's an obligate brood parasite. So this means that it has never made a single nest in its life, and it provides absolutely no parental care. So it shirks this responsibility onto other foster species or host species. So it's found a pretty creative solution to the insane costs of childcare, which the Europeans in the room might not be too familiar with, but the North Americans definitely are. But the host species aren't too keen on this. This is mainly because these little cuckoos are born with murder in their mind. So as soon as these little cuckoos are born, their first instinct is to really get in and shove those natal offspring out of that nest. So this effectively reduces the host's fitness to zero. So there's naturally you could see how this would lead to some coevolutionary interaction. So there's very strong selection on hosts to discriminate cuckoo parasites. So you really want to know if there's a cuckoo in the nest, while simultaneously there's really strong selective pressure on cuckoos to try to trick these hosts. So here you have a coevolutionary interaction between cuckoos and hosts. Now what makes this particularly interesting is that cuckoos are the ultimate parasitic generalist. So they're the parasitize more species of bird than any other obligate brood parasite. So within Europe alone, they parasitize over 100 species of hosts. So there's not just coevolutionary interactions between one host and one species of cuckoo, there's simultaneously interactions between the cuckoo and many different hosts all across Eurasia. Now you can use a lot of different life history traits to describe cuckoo parasitic success. So you could look at the host species that cuckoos are specializing on, you could look at their egg diversification, you could look at what natal habitats they're born in, and all of these traits likely interact to make cuckoos so successful at what they do. Because we're limited on time today, I'm going to just focus on one phenotype, and it's generally the phenotype that people really want to hear about anyways, but we're going to talk about this nice egg diversification within cuckoos. And I think you'll understand why in about 1 second. So interspecific diversity in common cuckoo eggs is pretty phenomenal. So here we just have cuckoo eggs within Russia, where on the left we have the cuckoo egg, which is much larger, and on the right we have the host egg. And this has captivated naturalists for centuries because there's very high fidelity between the cuckoo egg and the host egg, where they're even forging some of these very nice intricate patterns. So it's not just background color. Fortunately, this has brought a lot of field biologists out into the field to study these interactions. Because I'm more of a bioinformatician, so I rely on lots of field biologists across the world to send me nice blood samples. So whose shoulders am I standing on in terms of brood parasite genetics? So Claire Spottiswoode has a really nice paper looking at cuckoo finch genomics of eggs, where she found and postulated a pretty satisfying hypothesis wherein matrilineal inheritance of egg phenotypes can really perpetuate these systems. So you can think of it where if you have a cuckoo finch female who has an egg type, then she is able to mate with a male cuckoo finch of any egg type at all, but she'll always pass that egg type onto her daughters because it's on a matrilineal component like the mitochondrion. So then that daughter, because of host imprinting or something, will return the following year and always lay that same egg type. So that's kind of how we think this might be perpetuated. This was also postulated by Leslie Gibbs and colleagues in 2000 in the common cuckoos. And then Freude in 2016 on the right there looked at mainly microsatellite markers and saw some subdivisions within common cuckoos, but it seems a little bit murkier in common cuckoo, where at least a lot of variation is driven by this blue egg phenotype. Which brings me to my objective within this chapter, which was to see within cuckoo egg diversity, what are the matrilineal components? So in birds, where the females are the heterogrammatic sex, so they have a Z chromosome and a W chromosome, is completely linked with mitochondrion. So we wanted to see, is there maternal DNA contributions to here? Are there autosomal or biparental contributions to egg diversification? And do these things occur in allopatry or sometimes in sympatry? So to do this, I built a data set of basically every cuckoo I could get my hands on. So we have a resequencing data set of about 400 resequenced cuckoos. We generated a chromosome level assembly, and we made a quick matrilineal phylogeny there, just on the top, where we see about 7 major haplogroups. So on the left, we have those blue egg cuckoos, and this is a pretty ancient split. I'll show you a time-dated phylogeny later, but this split between the blue egg cuckoos and the rest on the right is several million-year-old. So what is the geographic distribution of these haplotypes? So here, the pie charts correspond directly to those haplotype colors, with the size of the pie chart corresponding to our sampling effort. So we see that there are many haplotypes that are spread all across the landscape. So we see that these haplotypes are pretty resistant to gene flow, and that there's something that seems to be maintaining these across huge distances. So the background color on this map, the grays to the whites, shows the autosomal population structure at k equals 5. So this is showing that the patterns of isolation by distance at autosomal biparental loci is pretty expected by distance. Of course, we can formalize that. So we can look at just Spearman's correlation between geographic distance and genetic distance, where we see a correlation above 0.9 for autosomal biparental DNA, whereas this really breaks down for these matrilineal components. So there's a few things that could be driving this. You could have balancing selection of these matrilineal genomes. You could have just female bias dispersal, which is also kind of the status quo in birds. Or you could have adaptive integration across different populations. Now that we've established that these matrilineal genomes are pervasive across the landscape and are maintained, what phenotypes are associated with them? So qualitatively, if we look at the egg diversity within each of these haplogroups, we see some patterns where some egg types are somewhat restricted within haplogroup, but there is a lot of overlap. So there's a few different ways that you can formalize the testing of this. The one that we're settling on mainly now is to look at do ancestral reconstructions on the mitochondrial tree and on the autosomal tree, and then see which tree requires the least amount of transitions in state for each egg type. So within this plot, we have the different egg types along the y-axis, and on the left side we have more matrilineal support. So this is more support for mitochondrial phylogeny for that egg type, whereas on the other side we have autosomal support. So we can see that there is not a pervasive mitochondrial support for many egg types, whereas it's mainly this E1 and E3, these bluish eggs, really seem to be associated with the mitochondrion, whereas we have egg types like E4 and E6 that seem to have more of an autosomal picture. But of course does not seem to be that clean. We can look at discordance in the tree. We've already seen quite a few of these plots today. But here I'm just going to highlight on those blue clades. So here we have ancestral reconstruction where the pie charts on each node show the likelihood of that ancestral state and egg. And I'm sorry if it's hard to discriminate color. My supervisor also is colorblind, and these plots are like a total nightmare. But with 11 egg types, it's a little difficult. But so the blue lines are a little thicker, which really shows that there is rampant mitochondrial autosomal discordance here. But it doesn't seem super clean. So really the next step that we wanted to take is, can we isolate a few different scenarios here and try to look at what candidate genes might be involved, really looking at fixed SNPs between some of these comparisons. So we leveraged 3 different comparisons, and I'll show a phylogeny here to bring this all back into context, but the 3 comparisons we wanted to look at are which mutations might underlie this transition between the spotted eggs, which seems to be the ancestral state, at least for the autosomal tree, and the blue eggs. Then within this blue egg lineage, we have a reversion into the ancestral state. So that's our second one, this reversion in orange. And then we have a contemporary diversification. So within the non-blue lineage of eggs, we have a lot of diversification. So it's looking to see what are the SNPs that are associated in this branch. So here's that time-dated phylogeny with the hypothetical point mutations and conferring those different egg histories there. So we have that ancestral blue mutation where the split is sometime between 2.5 to about 6.5 million years ago, and this is occurring on an absolutely homogeneous autosomal background where you have these ancient haplotypes. And then we have that reversion in the orange down there at the bottom, and on the right side we have the contemporary diversification. So these are the three scenarios that we'll then look at next, where if we look at the proportion of fixed SNPs, so on the y-axis here we have our mitochondrial and W components, so these are the matrilineal components on the top, the Z chromosome and then the autosomes, and the bar chart corresponds to the proportion of fixed SNPs that we assayed. So you can see the blue there is that ancient blue comparison, so this is the transition to blue eggs. We see that almost 75% of all assayed SNPs are fixed. So there's a huge amount of W chromosome and also mitochondrial SNPs that are completely fixed in this comparison, which of course makes it a little difficult to find out what genes are involved here. But we see absolutely no autosomal fixed SNPs. Even with the green color here, this is that contemporary diversification of the non-blue diversification from the spotted to the more the striped eggs. We see some mitochondrial and W chromosome contributions as well, but there are some Z and autosomal determinants. So if we only take these SNPs and then we say, okay, what are maybe the more functional SNPs that are involved here, we can look at which non-synonymous sites are involved. So if we only look at non-synonymous fixed SNPs in those three comparisons that are saturated for genes, we get an excess of oxidative phosphorylation, which is not too surprising because it is the mitochondrial. Many mitochondrial genes are involved here. But what was particularly interesting is this gene right down here at the bottom, NDUFAF4. So in all other birds, this gene is only on chromosome 3 or 4. It's an autosomal gene in all other birds. And in the cuckoo, we also see it on chromosome 3, but we also find it in many copies on the W chromosome. So this is where the spidey senses started tingling. And so then we downloaded all the HiFi and Illumina data for all other birds in the Cuculiformes family because we wanted to see if this gene also lies in the W there. So here on the y-axis we have our different species in phylogenetic order going from the cuckoo out, and then female and male coverage of this gene copy on the autosomal and the W copy. So we see only there in the bottom within the Cuculus genus that this gene, NDUFAF4, does seem to only be within Cuculus. And it does have the female-specific pattern of no male coverage. From a functional perspective, this makes sense. So in other birds, the blue egg is conferred by 3 different pathways, at least in chicken, quail, and duck. And the pigment that it relies on, biliverdin to make blue eggs, and protoporphyrin to make red eggs, both relies on heme. So heme is directly involved with oxidative phosphorylation, and it is very tightly linked to the mitochondrion. So we think what's probably happening is that cuckoos have co-opted the mitochondrion as the most rapidly evolving locus in combination with some autosomal genes that are involved with oxidative phosphorylation to confer just this huge armada of phenotypic variants in egg type. So it has really allowed them to become such a diverse parasitic generalist. But that is not the whole story of these matrilines. So we have some previous work with some folks in Portugal where we looked at female limited polymorphism. So in cuckoos, we have gray and rufous cuckoos, which is also a parasite that helps, a phenotype that helps them parasitize. And we found that within 2 different species, there's the same genetic locus on these W chromosome that goes back before a speciation event. So we found that these have been maintained in both species for over 1 million years. So these matrilines are under very strong selection, likely negative frequency dependent selection, and that cuckoos provide a pretty fascinating example to look at coevolution in the wild. And with that, I would like to say thank you to the Max Planck and to LMU and Evolution, and to my adopted family at the University of Victoria. And with that, I'm happy to take questions later. Just find me, and thanks for listening and coming.
So it's a coevolution session, so talking a little bit about coevolution and how we might use phylogenetic comparative methods to learn something about this process. And then some speculation on how does this fit in with diversification. And I'd like to give some ideas then how we might move forward using phylogenetic comparative methods. Okay, I got old, I can't see my slides anymore because they're so small up here. So I'm sorry if hopefully the captions will give you a better idea of what I'm saying. Okay, so I have 3 questions that I'm going to try to address in this talk. One is, what is coevolution? Really just like to define coevolution, get back to the roots, the idea that it's based on reciprocal selection, right? That's really one of the only forces that can be maintained through time when 2 species traits are being reciprocally selected. Then we have a good, all-encompassing definition of coevolution. Okay, how does that play into the idea of coevolution and phylogenetic comparative methods, and then just some ideas on coevolution and diversification. Okay, so a good way to define coevolution is using Janssen's additional definition. I know there's been a lot of work since then, but I still like this definition because it nicely captures the idea that coevolutionary change in a trait of one population affects the trait in another population, and in turn, that trait in the other population back affects the trait in the first population. It's getting at this idea of reciprocal selection, okay, natural selection. Scott Naismith has a nice textbook on coevolution. He has a nice quantitative genetic approach to it, for example, using the Breeder's equation, which some of us who are familiar with quantitative genetics, this might make more sense. A change in a trait value X, or mean trait value X, is equal to its heritability or additive genetic variance, as well as the selection coefficients, which in turn is a function of some trait Y. And then you have the reciprocal model below it, where Y is the changes in Y are dependent on the heritability and the changes in X. Okay, so that's a good model for microevolution. Quantitative genetics is good for microevolution. Not so convinced it's that good for macroevolution when we start heading over to phylogenetic comparative methods, and that's ultimately what I'm going to be talking a bit about today. Okay. Nonetheless, what we're left with is this idea that coevolution, one way we can define it, no matter which time scale we're talking about, is reciprocal selection always has to be there. That, okay, and I guess coexistence. You're not going to evolve towards each other unless you're coexisting. All right, so I'll introduce a classic example of a potential coevolution, and that's figs and their pollinating wasps. We all know there's something like 800 species of fig trees, and something like each of them has a roughly unique pollinating wasp, and we think they've co-diverged. There is a lot of host switching and all kinds of messy stuff going on, although some recent phylogenies have shown that they're actually pretty good species, at least on the fig side. There's less introgression than we previously believed. That's a story for another day. I would like to make the plea to the fig biologists in the room that we do need more data on figs. We just we seem to have stopped collecting data on figs for the last 20 years, and it's such a cool system, a classic system, that needs updating. Okay? How does it work? Well, we have fig wasps, they enter the fig, and they pollinate the fig. It's a mutualistic relationship, and they also use some of the uniovulate flowers in there to lay their eggs in. Okay? In order to do that, they need to insert their ovipositors down the styles. Do you see the mouse pointer over there? No? Okay. Do you see the red arrow? All right, that's the style length, right? The ovipositor, it's like roughly equal to the length of that little tail sticking out the back of the fig wasp. That's the ovipositor sheath. Those have got to match for successful oviposition. Why? Because the fig wasp needs to insert its ovipositor down there, stimulate the nucellus to get gall formation and to provide food for the developing larvae. If those two traits don't match in length, we're not going to get successful larvae production. Okay. Across species, we have variation in that trait. We have variation in style length, and we have variation in ovipositor length, and they tend to match pretty well. It's a good candidate for example, the phenotypic matching model if we were doing this in populations. And that is the basis of this idea that this is coevolution, right? So these two things are affecting each other. They're dependent on each other for their continued reproduction. And we think that it's natural selection that's causing changes in these two traits. Now the idea is just to test, is it actually reciprocal selection or not? Okay? All right, so I'm using this data set from George Veblen from 2004. Can you guys hear me? Okay, I'm probably going in and out. Right, so there's this tangogram here of these wasps. It's a really old phylogeny, as you can see. It's not very well resolved, right? And it turns out the phylogenetic structure actually makes quite a difference to what we're going to conclude from these models. But nonetheless, we do see some of at the bottom there especially, we see this codivergence happening in the fig wasps. There is a lot of codivergent structure. More importantly, when we look at the actual trait values on the left, that's just on the raw scale. We have one trait, I think it's ovipositor length on the y-axis and the style length on the x-axis. We see a very tight correlation between those traits, right? It has an R of 0.89, so an R squared of about 80% of the variance is explained by the regression between these two traits. So it's a strong, tight relationship. Okay? So Veblen applied independent contrasts to these data sets in 2004. He had a pretty clever way of doing it. He included body size, and the idea there was to show that actually the fig's style length is more dependent on the wasp's ovipositor length is more dependent on the fig style length than it is on the actual body size of the wasp. Okay, and it turns out, yes, that's the case, but when you apply independent contrast, you have a problem that you have an underlying assumption of a Brownian motion model. And if you have adaptive signal in one part of the phylogeny, what you're doing is you're removing that adaptive signal. With coevolution, you have adaptive signals in both phylogenies, so you're moving adaptive signals twice. And you can see that when the correlation or the R squared for the independent contrast, for example, went down to 0.4 if you apply independent contrast. Yes, it was still significant, but that's not, it's a massive reduction in power to deduce what we want to deduce. Is this reciprocal selection? And of course, a Brownian motion model cannot model adaptation towards optima, which is also not exactly modeling reciprocal selection. Okay, it's modeling a pattern, yes, and it looks like correlated evolution. We just don't know what's causing the correlation. Okay, so there's a couple of ways to model reciprocal selection. We can use the quantitative genetic way, where we're a little bit removed from reality, but at least we're incorporating additive genetic variation and selection coefficients. But if we're going to go to the macroevolutionary level, so far, I guess, at least in my opinion, the Ornstein-Uhlenbeck process is the best way to model selection. Okay, it's a rubber band process where the further you are away from some optimum, the theta value over there, the stronger the pull towards that optimum. If you have natural selection acting, you can estimate the rate of adaptation towards that, what we call a primary optimum. It's an average optimum of a bunch of species evolving in the same niche. That alpha parameter tells you that. And the change in y, that's the trait value, is dependent on this selective part of the model or deterministic part of the model, as well as the noisy part of the model, which is a Brownian motion, and that's basically the Wiener process. To the right of the deterministic process, if we set alpha to 0, this whole model collapses to a Brownian motion model. Okay? If we're going to do coevolution, we need a multivariate version of this. So we've been developing MV slouch, which is multivariate stochastic linear Ornstein-Uhlenbeck models for comparative hypotheses, which is becoming a bit of a mouthful. And all I've done there is I've replaced the A scalar value there with a matrix. It's an A matrix in this case, and then the E replaces the sigma. That is now a matrix. So we imagine we have two traits. We're going to have two alpha values on the diagonals. That's going to be the rate of adaptation of each trait to its optima. But we're also going to have off-diagonal values, and that's going to tell us a little bit about something about how these two traits affect each other while they're adapting to their optima. And we can use that to start estimating whether or not we actually have reciprocal selection. Okay? To see reciprocal selection, we want to see a model where both off-diagonals are parameterized. In other words, both traits are affecting each other's evolution, and they're doing it in such a way that it's adaptive. Okay. We can also discriminate whether this correlation between the traits is then happening in the deterministic component of the model or whether it's happening in the non-deterministic component or the stochastic component of the model, in which case it's vicariance or something else going on. And perhaps we have less evidence then for reciprocal selection. Okay, I just wanted to give a shout out to Mansau et al. They had a nice way of using these Ornstein-Uhlenbeck models and incorporating the phenotypic matching model of Naismith et al. I thought that was a pretty cool way to do it, but I don't think it's fully multivariate yet, so we were not quite there yet, having the ability to fit these models. Okay. How do we interpret these models? A little bit more on that. So I'm zooming in on the deterministic component where we have this A matrix, which gives us an idea of the rates of adaptation of traits towards their optima. In this case, the optima are set by whatever the other trait value is, right? Because we're modeling reciprocal selection. We're imagining a bibarrier trait. Imagine the simple fig wasps. We have the length of the ovipositor and the length of the style that they need to oviposit in. That A matrix can be represented by subscripts, A1, A1, and A2, A2 are the diagonals. That's the rate. If we were to estimate a model and we found something where all 4 of those squares were parameterized, we have coevolution, right? If only the diagonals were parameterized, we have independent adaptation to each other's optimum. They're not affecting each other's optimum. We don't have coevolution. We simply have evolution or adaptive evolution, okay? The mean structure of the model is the actual, it depends on, of course, the ovipositor lengths and the style lengths themselves that go into it and the variation in that across the different species. And then there are various types of models we can fit. We can fit an OU model. That's called an Ornstein-Uhlenbeck model. Ornstein-Uhlenbeck model, it's where both traits are evolving as an Ornstein-Uhlenbeck process, which will result in at least the potential of having all 4 alpha values. We can have an OUBM model, which is my baby. It's where one is an Ornstein-Uhlenbeck process and the other one is a Brownian motion process. Or we can have a BM model where both traits are evolving like a BM, in which case I'm going to get a move on. We can do a similar thing with the stochastic process part of the model, but there we're not really dealing with coevolution anymore. We're dealing with bicariance. Okay? I got 2 minutes, right? So these are called phase portraits of the alpha matrix. It's perhaps an easier way to describe the eigenspace than the actual models themselves. I borrowed these from MVMorph, where they do this for the sigma matrix, which is the Brownian motion model. It's got little arrows that point in directions. That's the direction of trait evolution. You'll see where the direction of trait evolution changes, we get those blue lines. Those blue lines are called null lines. If you see them orthogonal to each other, we have independent adaptation. If you see one of them is shifting off the perpendicular or horizontal, it means that one trait's driving the evolution of the other. And when they're both shifted off their perpendicular or horizontal, both traits are affecting each other. Okay, I haven't found an easy way to describe eigenspace yet, but this at least tells us which models we're dealing with, independent adaptation or co-adaptation. Okay, so it turns out my baby behaved the worst. Right, the OUBM model, it's like I just had to kill my baby. I felt very sad about it. The OU model performs really well, okay? We have lower triangular or upper triangular alpha matrices are the best models. So what I did was I used either the fig tree or the wasp tree as the model of coexistence as well as phylogenetic structure. And the model, we couldn't discriminate between whether it's an upper triangle or lower triangular A matrix, which means we don't know which trait is driving it. However, with just a little bit of manipulation of that tree, I found that the best model in that case is one where we have both off diagonals parameterized, which is pretty impressive given there's only 32 species and we're trying to estimate something like 12 or 13 parameters with these multivariate methods, but there's strong enough signal here to do it. Okay, so this is the impetus to go collect more data to make this real, I guess. Okay, is that the end? Yes. Thank you to my wonderful lab for being here. My suggestion is we collect more data.